Good evening. On behalf of the Stem Cell Foundation of Australia, welcome to the Future Medicine Healing the Heart event. My name is Megan Munsie. I'm a director of the foundation, and I'm delighted to be your host this evening. I'd like to begin by acknowledging the traditional owners of the lands from which we are joining this event tonight. I'm coming to you from the lands of the Bunurong people of the Kulin Nation. I pay my respects to their elders, past and present, and recognise any people from First Nations who might be joining us. Now, heart disease is Australia's biggest killer, taking the lives of 18,500 Australians each year. Plus, more than 2,000 babies are born every year with congenital heart conditions. The National Stem Cell Foundation is a charity with two key missions. One mission is to support the stem cell researchers doing evidence-based research. Our other mission, and the reason we're here tonight, is to provide the broader community with information about this emerging field of regenerative medicine so that they can make informed choices about their own healthcare. Tonight, we're going to hear from three of the stem cell scientists whose research we've supported and who are paving the way for new therapies for heart conditions. James Chong is developing stem cell treatments to repair injured heart tissue. Enzo Porrello is developing tissue patches to provide extra pumping power for hearts that need it. He's also creating heart tissue in the lab to solve the mysteries of the different inherited heart conditions that some children are born with. James Hudson is making mini hearts to help find and test new drugs that could be used to treat heart conditions, including damage caused by serious cases of COVID-19. So what we're going to do is we're going to hear short presentations from each of our speakers before having a Q&A session where, importantly, you have the opportunities to ask your own questions. Please do this by writing them in the Zoom chat box that you'll see at the bottom of your Zoom window, and we'll get to as many as we can. I'd also like to introduce you to two people that are helping out from Science in Public. Sarah, who's providing technical support for this event. And if you have any technical issues, please direct them to her via the chat function. And we also have Tanya online, who will be feeding your questions through to me and tweeting from the Foundation's Twitter handle, which is @OzStemCell. If you'd like to share your insights and comments, you can use the hashtag StemCells4Hearts, and that's a numerical 4. Also, note that this evening's event is being recorded, so we can share it online at a later date. So it gives me great pleasure to introduce our first speaker, Associate Professor James Chong. He's a researcher at Westmead Institute for Medical Research and the University of Sydney. He's also a cardiologist at Westmead Hospital. Over to you, James.
Thanks, Megan, and thanks to everyone who's joining us online, and for anyone who's watching the video recording later on. As Megan mentioned, I'm a cardiologist at Westmead Hospital here in Sydney, and I also run a basic science group at the Westmead Institute for Medical Research and the University of Sydney. So I'll just share my screen here and start the presentation. Great, hopefully that's coming through. If not, someone yell out and let me know.
Just presenter mode if you don't mind.
Thanks, Megan. Okay, excellent. So being a clinician, and since we're here to talk about heart health and how stem cells can play a role, I like to start these talks with a little bit of a case scenario to put things into context. And this is a real patient some years ago now. We'll call him Mr. P. And Mr. P had chest discomfort. Like many people in Mr. P's situation, he didn't think it was his heart. He put it down to indigestion, but after quite some time, he relented to his wife's urging, and he came to hospital, and I was called in the middle of the night to perform a coronary angiogram. That's what you're seeing in the video on the right of screen. So heart attacks are caused by ruptured plaques, and that stop blood flow to areas of the heart. And that's what you're seeing in the video. Hopefully you can see my mouse. This area down here is blood. Well, it's the dye tracing the blood flow down to part of the heart. But what you're not seeing here is another artery, which should be supplying a large amount of the heart of Mr. P. So fortunately for Mr. P, we're pretty good at dealing with this situation now. Like in most, all metropolitan areas in Australia, maybe not so much in more regional areas, but we have a situation where we can perform what's called primary angioplasty. It's where we open up the artery using a little balloon and leave a metal scaffold, put a stent in to hold the artery open. And that's what you're now seeing in Mr. P's heart. So you might be able to faintly see the stent here, but then now I'm sure you could see that the blood flows are restored to this area of the heart. But unfortunately for Mr. P, as mentioned, this situation had been, the artery had been blocked for quite some time. And so this is an ultrasound of Mr. P's heart. We're looking at the left ventricle. That's the major blood pumping chamber of the heart. Now, what you should be seeing is vigorous contraction like this area here, but you should be seeing that all around the left ventricle. And you can probably appreciate that, say, this area and all around is not really contracting like this area of the heart. So this means that the heart's not pumping blood around the body, and this causes a situation known as heart failure. And heart failure is a major problem. Unfortunately for Mr. P, he ended up dying during his hospital stay. And that's despite us being able to open up blood flow to his heart. Now, this situation occurs because the heart, like the brain, is one of the least regenerative organs in the body. Once it gets to this particular stage of heart failure, drugs have a reasonably limited effect. Now this is in contrast to when we are able to catch heart failure at the early stages, where drugs can have an important effect. But even then, there's often an incremental downward spiral. And once you get to the severe stages of heart failure, like Mr. P, really the only chance for cure, that's complete reversal, is a heart transplant. Because the drugs don't restore the pump function of the heart, they generally just halt or stop the decline of the pump function. But most of you will know that we don't really have enough donor hearts to go around. And in fact, we'll never have enough donor hearts to go around with our aging population, etc. So like Mr. P, that really only leaves a situation where palliation. Many of you might have also heard about mechanical hearts or left ventricular assist devices, and they're a very important part of our armamentarium now, but they also have their limitations. But we here on the call and many others feel that stem cells and the field of regenerative medicine can play an important part in feeding the unmet need to cure or at least to treat these severe stages of heart failure. Just to talk a little bit about heart disease, it increases with age. It affects males more than females just a little bit, but as you can see from the graph on the right, it really only perhaps protects females just by a few years, really. And many people don't realise this, but at the severe stages of heart failure, there's a very sinister prognosis, probably at least as bad or worse than many forms of cancer. So that it's thought that at the later stages of heart failure, that's class 4 heart failure, it's quoted that patients often die within 1 year of diagnosis. Now, this is one statistic. It's clearly not always the case. And obviously this costs our health system quite a lot of money, but probably more importantly, it causes poor quality of life for those suffering with heart failure. So as mentioned, James and Enzo feel that stem cells can really do an important, have an important part to play in curing heart failure. And if you're on this call, then you'd know a little bit about stem cells, but I might just take the time just to explain at a very rudimentary level what a stem cell is. And at its most fundamental explanation, a stem cell needs to be able to regenerate itself, and it needs to be able to go on to form a different type of tissue. And for us, it's making heart muscle cells to restore that pump function of the heart. So you can see how those two things can combine to become a very powerful therapeutic option. Because it can replenish itself, we now potentially have an unlimited supply of heart muscle cells. Now, contrast that to that situation where I said that donor hearts are in short supply and will always be in short supply. So this is an example of what we do in the lab. This is images of real heart muscle that we've created from induced pluripotent stem cells. And you can see that they're spontaneously beating in the dish. So this is a method that we're trying to use therapeutically, and we can grow these now in the lab literally by the billions. And in fact, we anticipate treating a heart patient with many millions of these cells. And to generate that scale of cells, we need new technology, and the technologies come really in leaps and bounds. So the images on the left are probably from about 10 years ago now. When I was in Seattle, you can see that there's not small petri dishes, they're already large flasks, but that was a very onerous process. Whereas now with the University of Queensland colleagues, Professor Peter Gray and Andrew Prowse and their team, we're using bioreactor technology to really upscale this clinical manufacture. So I just want to leave you with a little cartoon video just trying to illustrate what we're potentially aiming to do with this stem cell derived heart muscle. So this is just showing what I said about heart attacks causing lack of pump function into the damaged heart, and that causing heart failure. And what we hope to do is to come in and then replace the injured heart muscle with these new stem cell derived heart muscle. And you know, hopefully in as a potential possibility one day, who knows, in the future, we might decrease the need for heart transplants, or maybe even completely overcome it. So with that, I'd just like to thank you for your attention. These are the people in my group who are working to do this research, and thanks to the Stem Cell Foundation for the ongoing support. So I'd welcome any questions.
Thanks so much, James. And that was a fabulous presentation. And you know, your work is so impressive. I'm just wondering whether anyone has any questions. If you do, please pop them in that chat box, and Tanya will let me know what they are. But I'm just going to, probably got time for one now, and we can come back, James, a bit later on. But you know, you mentioned that you can create billions in the lab, and for each patient you need millions, perhaps potentially of these cells derived from stem cells. But when do you have to put them in? Like you talked about sort of the acute timing. So what, you know, how hard, you know, do you have some wriggle room there, or must it be done in an acute situation?
Yeah, that's a great question, Megan. And to be honest, we don't exactly know. You know, when you're looking at groundbreaking medical technology, it undergoes many iterations. You look at bone marrow transplantation, whole organ transplantation. They went through a number of protocol changes, let's say. So, but we have well, at least our approach is that the patients that would need it will have heart failure established. Now, whether they are the best patients to receive these heart muscle cells. The future will tell, but that's our approach. We feel that they're the best candidates to receive the initial efforts. I should also point out that although I used heart attacks as the illustrative example in my talk, I did that because heart attacks are the major cause of heart failure, but we envisage possibly that this therapy could be used to treat a range of heart failure cases from any number of disease etiologies. So long answer to your question, it's we don't exactly know, but the first steps will be in patients in our group, in our hands, we'll approach people who have had heart failure for some time.
Fantastic. Well, thank you so much. I'm going to invite you back after we've heard from the other two speakers, but thanks so much for kicking us off. And I really appreciate that slide on a bit of background about stem cells too. That was really helpful. So thanks very much, James.
Sure.
Now it's time for our next speaker, which is Professor Enzo Porrello. Enzo is the theme director of stem cell biology and head of the heart regeneration group at the Murdoch Children's Research Institute. Over to you, Enzo.
Great. Thanks so much, Megan, for the introduction, and thank you to the Foundation, National Stem Cell Foundation, for hosting tonight's event, and thank you all for attending. Looking forward to the discussion. So tonight I'm going to talk about the research that we've been doing in our lab over a number of years, developing bioengineered heart tissues from stem cells, and how we're using those bioengineered heart tissues both to understand the causes of childhood heart disease, but also as potential regenerative therapies for children who develop severe forms of heart disease and heart failure. And really my motivation for this research stems back to my own family's personal experience with childhood heart disease. This is a photo of my two cousins, Frankie and Peter. And this photo was taken in the 1970s. Both Frankie and Peter were born with a relatively common form of congenital heart disease. They were both born with a hole in their heart. And fortunately for Peter, the hole in his heart healed over time, and he's gone on to live a healthy and full life. But sadly, his brother Frankie passed away at the age of 9. Today, the outcomes for children with heart disease have dramatically improved over the last few decades. And for the first time in history, in 2022, there are actually more adults living with congenital heart disease than there are children born with heart defects. And this is thanks to really tremendous advances in the surgical management and clinical management of these conditions over the last 30 or 40 years. However, these residual anatomic lesions in these survivors of congenital heart disease place these individuals at increased risk of developing heart failure throughout their lifetime. And despite these really remarkable advances in the surgical management of childhood heart disease, in 2022, childhood heart disease still remains the most common birth abnormality in children, affecting up to 1 in 100 live births. It still remains a leading cause of infant death in Australia and much of the developed world. Again, thanks to these surgical advances, these children are now living into adulthood, but this is creating new challenges, and these are really now becoming complex chronic diseases that require lifelong treatment. So our laboratory is really harnessing the power of stem cells to both understand the causes of childhood heart disease and also to develop regenerative treatments for these conditions. And thankfully, James Chong has already provided a very nice introduction into stem cells and pluripotent stem cells. And our laboratory also uses this technology. And one of the powerful things about this technology is that we can make these induced pluripotent stem cells from any individual. So we can take a blood sample, for example, from a child with heart disease, we can turn that individual's own cells and reprogram those cells into this type of stem cell, known as induced pluripotent stem cell. And those stem cells have the potential to differentiate into all of the different cell types in the body. And our laboratory, in collaboration with James Hudson, who you're going to hear from next, have developed methods that allow us to differentiate these induced pluripotent stem cells into three-dimensional beating heart tissues. And we can use these bioengineered heart tissues for a variety of applications, including disease modeling. So to understand the causes of heart disease, in particular, we're interested in inherited forms of childhood heart disease in our laboratory. We can then use these disease models as a platform for drug screening and therapeutic development to try and identify new treatments for these conditions. And finally, we can use this technology of tissue engineering to develop large engineered heart tissue constructs that can help to support the function of the failing heart. And so just a quick warning before I play these videos. These videos will show some open chest procedures with engineered heart tissue implantation in an animal model. So if you don't want to watch these medical videos, you should turn away now. But what you're looking at here is our pediatric cardiac surgeon, head of cardiac surgery at the Royal Children's Hospital, Christian Brizard, implanting one of these engineered heart tissues onto the heart of an animal. And then these patches are implanted and sutured onto the heart. And you can see here the heart beating away with one of these human engineered heart tissues implanted over the surface of the heart. And we hope that these tissues will serve as a biological assist device to help support the pumping function of the failing heart, in particular in children with heart disease. And we're now evaluating the safety and the effectiveness of this technology in animal models before progressing to human trials. So I'll end there, and thank you for your attention, and happy to take any questions.
So thanks so much, Enzo. And also I appreciated the warning. I think that was really nicely done. That can be kind of gruesome, some of the surgery, but still very impressive in itself. So we've got a question here that I'm going to raise now. And it's from David, who provided this actually before, this question before today's presentations. So he did this upon her hearing about the forum. So David has hypertrophic cardiomyopathy, or HCM, and he's noting that it's a hereditary disease with several known genetic alterations, and he's interested to know more about research that relates specifically to this condition. Is this something that you have been looking at the MCRI?
Yeah, so this is one of the forms of cardiomyopathy and inherited heart disease in children that we are interested in, and that we've been studying in collaboration with David Elliott's group at MCRI. And we again take advantage of the power of this induced pluripotent stem cell technology, which allows us to derive stem cells that are a genetic match to the individual that has hypertrophic cardiomyopathy. And so we've been using this approach to make heart muscle cells and then to understand the genetic causes of hypertrophic cardiomyopathy in children. So we can go in using technologies such as CRISPR-Cas9 gene editing. This is a method that allows us to go in and very precisely edit the genome of the stem cells. We can correct the underlying gene mutation that we think might be causing heart disease in that individual, and then we can model their heart function in the dish. And what we often see in these individuals with cardiomyopathy in our stem cell models is that their heart muscle beats irregularly in the dish, in the stem cell derived heart tissue, and it recapitulates features of the clinical condition that is seen in those patients. So we can use this as a really powerful model to mimic the clinical condition in a laboratory dish, and then to study the mechanisms that drive that disease and ultimately try to develop treatments for it.
I suspect this sort of disease in a dish is sort of perhaps more powerful than animal studies. It must be hard to kind of replicate some of these conditions in an animal model. So this would give you, I suppose, new insights.
Yeah, so I mean, one of the challenges with animal models is that they often don't recapitulate the human condition. And we know for many of the mouse models have been very powerful for studying cardiomyopathies, including hypertrophic cardiomyopathy. But for certain gene mutations that we know are associated with disease in humans, the mouse models don't fully recapitulate those conditions. So we really need new models and better ways of understanding human biology. And that's something that these human pluripotent stem cell technologies really provide us with, is a human model of human disease.
It's really interesting. And look, before I let you go and we go on to James Hudson, we've got a question here from Michelle, who asks, is there a way to implant stem cells without open heart surgery?
That might be a better question for James Chong, who's an interventional cardiologist and might be able to talk to some of the not less invasive methods. But I think for the applications that we're looking at in terms of bioengineered heart tissues that need to be implanted onto the heart, that's really going to require invasive open heart surgery.
Let's come back to that when we come back to the Q&A, because I think it's a really important issue, and perhaps James can talk to us a little bit more about when and why. That would be really helpful. But thanks so much, Enzo. That was a really great presentation. I'd now like to move on to our final speaker, Professor James Hudson. He's a group leader of the Cardiac Bioengineering Research Group at QMR Berhofer Medical Research Institute in Queensland. Over to you, James.
Thank you, Megan, and thank you to Tanya for organising this event today. And it's great to share some of our work with you here. So my work's going to talk about finding some using stem cell derived tissues in our lab to find new drugs. So not just implanting those cells, but using those as a model. So both James Chong and Enzo really nicely showed that we can use these human pluripotent stem cells to create the cardiac muscles in a dish. And over many years, we've developed protocols where in a 2-week period, we can create these cells. And then we've developed new protocols where we put these into 3D devices using engineering, where essentially what we can do is make these cells to a similar maturity of a newborn sort of heart in only a 4-week period. So we've identified the key signaling pathways and environmental stimuli that enable us to really accelerate heart development and have something that more closely mimics the way someone's heart is performing. As you could see in the video there, these tissues contract, and by measuring the deflection of the post, what we can do is actually record the forces that are generated by these tissues, which is essentially a measure of heart function in much the same way that James Chong showed in his really nice video of the ejection fraction in someone's heart. So some of our work has focused more so on inflammation-driven heart failure so far. James gave a really nice overview on how the heart can get heart failure from a heart attack. Enzo presented some nice data how genetics can cause heart failure. But there's a lot of data now suggesting that inflammation over your lifespan can be a leading predictor of heart failure later in life. And you may not have even had to have had a heart attack. And you'll note that this typically occurs in that older age bracket that James was talking about earlier. And over the next 10 to 20 years, this population is going to greatly increase. We're not going to be able to give all those people transplanted hearts once they start failing. And also, there's different forms of heart failure where we don't have as good drugs to treat them like we do with heart failure after you have a heart attack. And so one of those that's come to light more recently is COVID-19. We know from assessing millions and millions of people worldwide that your risk of cardiac injury increases every time you get a COVID infection. And some people have had up to 3 now, maybe even more. And each time it increases your risk. Your risk can be up to 4-fold higher. Now, COVID's just one aspect, and it's because it's been intensely studied because of the current pandemic. But there's also other viruses and things over your lifespan that can greatly impact your heart tissue. Luckily, the COVID-19 virus doesn't directly infect your heart tissue like some of the others, which just destroy your heart muscle. And this is the pathological effects from COVID-19 are much more likely to be mediated by the severe inflammation in your lungs that you get. What we did using our organoid system that I just showed you was we actually identified which of these inflammatory factors in your lungs are causing the dysfunction in the heart tissues. And we used a whole variety of different discovery approaches and biomedical research tools to actually identify what exactly was going on inside those cells. This enabled us to identify a number of drug candidates that could correct this phenotype, and we tested those in animals infected with COVID-19. One of these drugs, really luckily, was already in a phase 3 clinical trial in Canada. It's a drug called apabetalone, and they could rapidly repurpose it straight to a phase 2B clinical trial for people that get hospitalized with COVID-19. This is not yet available in Australia because it's pending positive data from the clinical trial in Canada. That's where the drug company is located. And hopefully if there's promising results, that can be given out more widely. Now, because this mechanism is not just the same in COVID-19, it's also applicable to other forms of infection. One of those that people might be familiar with is sepsis, where you have a bacterial infection or a viral infection that's quite severe. This can be a very severe form and affect your body quite severely in hospital. A lot of these patients end up in ICU. And we've developed a collaboration here with the Royal Brisbane Women's Hospital, where we're also looking to try and apply these drugs to people that have sepsis to try and improve their outcomes as well. This is an overview of one of our tissues we create, just to give you a bit of an idea of what it actually looks like down a microscope. So this entire tissue is 1 millimeter across. In white are all the cardiac contractile cells, and this is just fixed in place so that we can image it and stain it. In blue is all the cell nuclei, and in green and red are the other cells that help support the heart tissue. These other cells are very, very important for the heart tissue function. And researchers across the globe, including James Chong, have identified potential therapeutic candidates that target these other cells as well. We've recently just identified, and this is currently unpublished data, that these other cells are instrumental in these disease processes. This graph that I'm showing you here is basically the levels of a certain factor in people's bloods called endothelin-1. And this increases as people are hospitalized or on ventilators with COVID-19. And this also directly correlates to various outcomes such as acute kidney injury, myocardial infarction, and death. What we found in our tissue sort of format where we could test these is that there's a bunch of FDA-approved drugs that block these receptors. They're called bosentan, abrasentan, and macitentan, and they're used for patients with pulmonary arterial hypertension, which is a form where you have really high blood pressure in your lungs, and this can also occur with COVID-19. In this graph here, what we have is essentially stiffness of the heart. This is we see a stiffening of the heart as the phenotype rather than a lack of contraction. And these drugs can also completely abolish this effect and might be very good, useful tools in the clinic for this form of heart failure as well. And so I hope that gives you a bit of an overview of how we can also use these tissues for drug discovery. And on this slide, what I wanted to show is really, it requires a lot of work from many people across Australia, and it's really important that we continue to fund biomedical research in our country.
Thanks so much, James. So it's time for your questions now. So I'd welcome people to put their questions in the chat. And just because we know it's a bit confusing having two Jameses, please refer to James Hudson as Hudson, because that's apparently what his friends call him. And he's our friend too. So please refer to any questions for Hudson as such. And I'm just going to, before I invite our other speakers back to join us, Hudson, I just wanted to talk a little bit more about your work and you know, such tantalizing kind of discovery mechanisms and tools you're using to test or repurpose drugs to help people with COVID-19. And you sort of said it was in a trial at the moment. I'm just kind of curious about what the kind of next steps are and how long it might take, because this drug's already been approved by FDA for other applications. Yes. Is that right?
Yeah, that's correct. So the drug's been in development for a very long period of time. I think, 15 years or so now. And they've identified that it's quite effective in treating certain forms of heart failure already. And it was in phase 3 trials, mostly for people to stop their blood vessels from becoming inflamed and causing that heart attack that James Chong was talking about earlier. And so they have a lot of clinical data for, up to 3 years in 3,000 patients, and the drug's quite safe. And so that meant that the FDA could rapidly approve a phase 2 trial to look at whether the drug was effective for COVID-19, because they already knew it was safe. That's now being tested on 100 people and 200 people. 200 people get randomized into 2 different conditions where they receive a placebo or the drug. And this clinical trial has been going since the start of this year. It's due to finish now, and the results should be posted anytime soon. No one's allowed to know what that data looks like at this stage, because it's a really rigorous clinical trial to assess whether the drug works. So once that data gets posted, the FDA will review that, and if it's positive, they'll approve a phase 3 trial, which is a larger trial, which might also involve more sites like in Australia. And now, this drug's could be really effective for our pandemic, because it's more for people, it's not preventing the response. So it's not like a vaccination. It's more when you're really sick, whether you're vaccinated or not, you go to hospital and it helps you recover in hospital and prevent you getting worsening outcomes. So it's another tool that we can use. Yeah.
Oh, that's fantastic. So that's what I was going to ask you, was I was curious about when you would get access, if the drug was approved, when it would be applied. But it sounds like it's going to help people perhaps not be in hospital for so long and maybe not die.
Yeah, that's right. Yeah, and also hopefully prevent the detrimental outcomes. The mechanism of action will theoretically perhaps also work on some of those other organs like the kidney, and so it's trying to prevent that sort of multi-organ impact that you get with severe COVID-19. Yeah.
Oh, that's fantastic. Well, thank you so much for a great presentation, Hudson. And I'm going to invite Enzo and James back to join us now, and again open to questions. And I've got one here. Tanya's pushed through straight away for James Chong. Harry asks, would there be any tissue rejections for using stem cells? So would the stem cells be rejected, James?
Thanks, Harry. That's a great question. Probably the initial trials will need immunosuppression, because they won't be from the cells from that host. When this field first started, we were thinking that you could actually make induced pluripotent stem cells from the patient themselves, and then use that those cells to recreate heart muscle, etc. And in that case, there may not be cellular rejection, but at the initial stages, that other patient-specific option will be too costly, at least in the near term. So yes, we will need immunosuppression initially.
And is the immunosuppression that you would use very similar to what people who had, say, a heart transplantation or a kidney transplant? Yeah.
Yep, that's our approach. We've modelled our immunosuppression regimen on what would be used for a whole heart transplant. However, we feel, as I talked about, that constant iteration in medical technology. So we envisage if the antigenic tissue, that's the tissue being rejected potentially, is slower than a whole heart, then the immunosuppression could be wound down as well. I mean, that's what we think, but we'll need trials to prove that.
Yeah, you have to just this constant evaluation, isn't it? And rethinking as required. Well, that's great. And now I've got a question from Alan for you, Enzo. Alan asks, will the stem cells displace the damaged heart, or will they layer over them to assist?
Yeah, it's a really good question. So our approach with the heart tissue patches would be to layer that patch over the damaged area. So it would essentially form a layer of heart muscle that lies over the defective part of the heart muscle to help it to pump more rigorously. So we wouldn't be replacing that underlying damaged tissue. The engineered tissue would sit over the heart and help to support the heart's function.
That's very interesting. And James, Faye is asking, how is this different from any other similar work that's been done before, and I suppose also done overseas? And have you had any regulatory challenges?
Yeah, Faye, that's a great question. I think the major thing for regenerative medicine is that for heart disease, or for heart failure in particular, and for congenital heart disease like Enzo is working on, is that we're really addressing the underlying pathology and replacing the loss of force-generating units. A lot of our current small molecules or drug therapy is really trying to dampen the decline. So that's probably, in my mind, the biggest shift in what's come before. Now, regulatory challenges, yes, there's a lot, and we're working with the Therapeutic Goods Administration to work through various regulatory approvals, which are needed to keep people safe, and that's important of regulators and well-designed clinical trials. So there are a number of hurdles, as with any new technology.
Did you want to elaborate at all on some of those hurdles? Like, what do you think is the most significant of the challenge that you face for to advance your technology? Just a simple question, James.
Okay, well, one of them that has really concerned people, and I think there was another question in the chat, is how you can control a stem cell, a pluripotent stem cell, and make sure that it's not going to become other types of cells that you don't want, and then go on to potentially form cancers. And really, the approach that many people have taken is that the differentiation protocols have got better, and that the detection of other, let's say, contaminating cells gets screened out by various processes that each individual group and then the field will be using, so-called release criteria. I mean, that's just one, but probably a big one that are on people's minds. Another one that our group's working on is heart rhythm abnormalities that can come from these transplanted cells, and our group has shown that a combination of drugs can very significantly suppress it. And we're also found that particular cells, we think, are causing these heart rhythm abnormalities. They're not ventricular, these pumping chamber cells, but they're more top chamber atrial cells or pacemaker cells. And so we're working at approaches that can take them out either by cell sorting or by changing the way we push the fate of the stem cells.
So this issue of getting the right cells at the right number in the right place at the right time is going to be sort of the technical as well as possibly even the regulatory challenges, right? So associated with some of these things. And I was just going to turn back to Hudson, because I remember from your presentation, you did talk about the fact that you have been able to optimize almost like the maturation. So you've been able to bring these cells up quite quickly. I think you said a 4-week period to, in quite a short period. Maybe you can clarify it for me how long that takes. But they were sort of more like a young child's or a baby's cells. And I'm just going to ask you a little bit to reflect a little bit about this issue of maturity. So what we create in the dish, what we can produce outside the body, how representative is it of an adult's heart cells, for example?
Yeah, it's a really good question. It's a big issue in the field for making any type of cell from pluripotent stem cells that people have had to deal with, whether that's the heart cells or the neural cells, and trying to use those to model the system. In some cases, like in some of James Chong's work, it might actually be more beneficial to not have them fully mature, because you want those cells to be able to integrate into the heart. And if you have a large, stiff, rod-shaped cell, it's not going to have the same integration and survival sometimes. And so different attributes are important for different types of uses of these cells. In our particular case, to more closely mimic the heart, what we need to do is try and get those as close as possible, and that's why we've worked on those protocols quite substantially. To date, we've done a lot of testing, and they are quite representative. There's still a few things that are different, but just as a perfect example, we can put on adrenaline and all the various things that might make your heart rate go up, or might make them contract harder. Like when you go for a run, your body releases all these hormones. And we see those exact same effects in our system as well. So we always got to be mindful of the limitations, but I think it's a pretty good model to get the screening to more quickly identify drugs that still then need to be tested in animals and humans.
No, that's very helpful. Thank you, James. And I've got Hudson. I've got a question here from Nicole. And it's not to anyone specifically, but she's asking, are there any plans in the future to use stem cells collected at birth for heart regeneration? So Enzo or James Chong, would you like to make a comment about that?
I think this sort of goes to the idea that James Chong mentioned earlier on about induced pluripotent stem cells and the idea of a personalized replacement cells for an individual. And in the setting of congenital heart disease, I think that the long-term goal, and I guess the holy grail of this sort of approach, would be that you'd be able to make the stem cells from that child at birth, when they're initially diagnosed with their congenital heart disorder. You'd be able to engineer a heart tissue patch that's genetically matched to that individual, because you're using their own stem cells. And because those children are often undergoing a series of operations during the first few years of life, you could potentially have time to make that tissue in a dish that's genetically matched to that individual, that could then be implanted back into that individual to support their heart function. And in that scenario, you would have a situation where you'd have a tissue that was genetically matched to the individual and that would avoid complications associated with immune rejection. But this is really at the moment not really feasible, and the cost of cell production and a lot of the regulatory hurdles sort of really make that not an option right now. And so the approach that most people are taking are to use off-the-shelf clinical grade pluripotent stem cell lines that would then be used to produce the tissues, but you would then need immunosuppression following transplantation of those tissues.
I think sorry, is that James Hudson?
Both Enzo and James have both so like you both talked about the immunosuppression issues, right? And there's new technologies becoming available where you have these sort of immunocloaked stem cells, but by really intimate knowledge of how immunology works, people can start to modify those cells so that they're not recognized by the immune system. What are your views on those, like to potentially overcome some of these issues?
Okay, I think if that can happen, that would be a game changer. It's a tricky thing, as you know, Hudson, but it's an intense area of research, and that would be great.
This is the idea of the universal donor, isn't it? So being able to have a stock that we could then differentiate into any type of cell for any person. So yeah, I think it's great for us to talk a little bit about that idea of personalized medicine, because I think it gets a lot of airtime, but as you've all mentioned, there's some challenges, right? So, whilst it means that it may overcome rejection, there's a big challenge around getting a quality product. You have to do every time you want to do the testing, you have to do it for that person, and that does really become a feasibility and of course a cost issue. But it's sort of hard to wrap my head around how that would work. But this universal donor sounds really interesting. How close do you think we would be to having a source of cells that would be like that? It's still quite a way away, isn't it?
I think there are a number of studies that have shown that it's feasible to make these universal donor lines that can evade the immune system, and so that it's a very active area of research. But there's still a lot that needs to be done in terms of evaluating the safety of that approach, and there's a lot of things that we don't know, and the immune system's complex. So I think time will tell, but those studies are ongoing.
I've got one final question. Oh, sorry, James.
Megan, I would just say, just about the personalised medicine and dreaming big, which is what the foundation encourages and what we do here. It wasn't that long ago that we were struggling to sequence the entire human genome, and now we can do that incredibly rapidly, and it's changing rapidly how we view genetic diseases and possible options. So, never say never with personalized medicine and these further down the track options that we're discussing today.
I think it's a fabulous way to finish, and I can see Enzo would like to make a comment.
I was just going to say, related to that, I think also the cost of cell production is coming down, and we're finding cheaper and cheaper ways to make these cells and to industrialize the technology. So I agree. Right now it's not possible, but that who knows what will be possible in the next 5 to 10 years. I mean, 10 years ago, what we showed today was not possible, not even conceivable. So yeah.
I think
also,
it's a beautiful
the
way
diagnostics.
To
Oh, sorry.
You go ahead. Hudson.
Also, James was touching on all the different regulatory things, and all the diagnostics to do those things have advanced substantially too. So, to prove that there's no remaining cancerous, potentially, tumor-forming pluripotent stem cells in there, we can readily do that now with our new technology, and overcome a barrier that was a massive barrier previously is now very simple for us to assess. So there's so many elements into that personalized medicine question, not just in the stem cell field, but more broadly as well.
I think this dream big is a fabulous way to finish the formal part of tonight. And I just got a few words to say, but I think it was a really great conversation, and I'm very sorry for the additional questions that we have coming through the chat that we haven't had time for. Hopefully we can get back to you and provide some responses, but thank you so much, everybody, for absolutely fantastic questions. And if you'd like to now join me in thanking our magnificent panel, James Chong, Enzo Porrello, James Hudson. This is the first of the Future Medicine series of public events that we'll be hosting. The next event will be a live event in Melbourne in November at the State Library, where a different panel of experts will discuss emerging cell and gene therapies, such as new treatments for children born with the rare inherited condition spinal muscular atrophy, where the nerves that service muscles don't work. And this is a gene therapy that is aimed at reversing that. We'll also be speaking about research that the Foundation funds at the Centre for Eye Research Australia into gene therapies to save sight for people with retinitis pigmentosa and other conditions that affect the retina. So we'd love you to join us for that event if you can. We'll also be doing a recording, so you could listen to it, watch it some other time. I'd also like to mention that if you or someone you know has any questions about stem cell treatments that are currently available or about clinical trials, we've got a new information resource that might be of interest and could help. The Foundation has a guidebook called What You Need to Know About Stem Cell Therapies, and it can be downloaded from our website. And the URL is www.stemcellfoundation.net.au. And of course, while you're there, if you'd like to make a tax-deductible donation to the Foundation, that would be lovely. We use donated funds to provide public and patient education to support emerging researchers. And in fact, our three speakers tonight are all past winners of our Metcalf Prize and each received $50,000 to support their careers. And I think you'll agree that that was money very well spent. Other scientists we've supported are working on everything from fundamental research to improving treatments for cancer, MS, cystic fibrosis, and as I mentioned, vision loss. If you want to read anything more about their work, please visit our website. There's some profiles there about the researchers that we fund and the projects. And of course, if you'd like any more information about making a donation or about the Foundation, please contact our General Manager, Graham Megan, via the inquiries email. That's inquiries@stemcellfoundation.net.au or via the contact page under the About tab on our website. So thank you again to our wonderful speakers and for all of you for joining us tonight. And for those of you who are listening at a later time, thank you so much for joining us and have a great evening.